The next thing we're going to talk about is the cell cycle. Again, this is a lot of information to cram into just a couple of PowerPoint slides. I apologize for that. I really feel like the book should have expanded on this information a little bit more, but we're going to run with it, okay? So, a cell is going to be doing its regular cellular activities in what we consider a G0 phase. So let's think of a skin cell, just because it's an easy one to think about. So when a skin cell is sitting on our skin, being a skin cell, it is not actively dividing, it is acting as its other purpose in life is being a protective skin cell. It's sitting in a G0 phase. When something happens, whether it be an injury or just natural cell death of a neighbor, something could happen in order to trigger that cell to go into a cellular division phase, okay? When that cell is triggered to divide, it is going to enter cellular division, okay? It's going to enter what we call the cell cycle. There are multiple different phases of the cell cycle, and that's what we're going to talk about now, okay? So when a skin cell is being a skin cell, a muscle cell is being a muscle cell, whatever it may be, it is in what we consider G0, so not a part of the cell cycle. When it gets triggered into the cell cycle, the first phase is G1, okay? It's going to go from G1 to S phase, from S phase to G2, and from G2 to M. M is going to be mitosis, and we're going to get to mitosis, okay? So let's talk about the genetic material, because that is the most important part of the cellular replication during cell division. We need to duplicate the genetic material so that each new daughter cell has the same genetic material that its parent cell had, okay? So we go through DNA replication in order to make a homologous pair of chromosomes, okay? So homologous pair meaning same pair, matching pair of chromosomes. So remember that chromosomes are the packaged genetic material, okay? So the genetic material can totally wind up and bind together during cellular division in order to make these pretty packages, okay? These pretty packages is the sister chromatids that are going to be wound up on a kinetochore. And so two homologous chromosomes are going to be then bound by the centromere. Lots of new terms, I know, just roll with me, okay? So each pair are then going to be separately inherited from different parents, okay? And this DNA replication is going to happen. Okay, let's talk about cell division, which is mitosis. Mitosis followed by cytokinesis. So when I was in school, we only had 4 phases, and we memorized those 4 phases as PMAT, prophase, metaphase, anaphase, and telophase, and then followed by cytokinesis. Okay, since then, they've added this additional phase called prometaphase. So there are now 5 phases of mitosis. Mitosis is cellular division, and it is the division of the genetic material into 2 nuclei and 2 sister cells, okay? So in prophase, when you are seeing prophase, you're going to see the condensing of the genetic material. So the genetic material goes from spaghetti-looking like material in the nucleus to the dense chromosome shape, okay? You're also going to see spindle fibers emerge. These spindle fibers are going to emerge from the centrosome. And then you're going to see the nuclear envelope, so the nucleus, is going to dissolve its nuclear envelope, and that's going to allow for easy access of the chromosomes. And the last stage of prophase is the centrosomes are going to move towards opposite poles. Okay, we'll talk about what the centrosomes do here in a second. So in prometaphase, chromosomes are going to continue to condense. The kinetochore is going to appear at the centromere, and the mitotic spindles are going to attach to the kinetochore. So the centrosomes and the kinetochores and the mitotic spindles are all materials that are going to be involved in pulling the sister chromosomes apart. Okay, so the next stage we're going to go on to is a metaphase. Metaphase I think of as meat in the middle. Okay, so what happens in metaphase is that those chromosomes are all going to line up on the metaphase plate in the middle. So each sister chromatid is attached to a spindle fiber in the middle, and they are going to be attached at opposite poles. Okay. The next phase is anaphase. I think of anaphase as a part, because what's happening is that the sister chromatids, now called chromosomes, so each new cell will have a chromosome. They're going to get pulled apart towards opposite poles by the centromeres and the chromatids. Certain spindles are going to elongate. Okay, so anaphase, the chromosomes are pulled apart. The last phase is telophase, and cytokinesis kind of run together. Okay, so in telophase, the chromosomes are at opposite ends, and they start to decondense back into their regular spaghetti shapes. The nuclear envelope is going to surround each chromosome, again, each set of chromosomes again. The mitotic spindles will then break down again because we don't need them anymore. And the spindle fibers will continue to push the poles apart in order to divide into new cells. The last stage, the last thing to happen, is called cytokinesis. Cytokinesis is that final pinching apart of the plasma membrane, going from one cell to two sister cells. Okay? In animal cells, we consider it causing a cleavage furrow during cytokinesis. In plant cells, they are not so flexible. So in plant cells, they are going to create a new cell wall down the middle to separate those two daughter cells. Okay, that is a lot of information. We do have an entire lab over this, and that will help you guys understand. So mitosis is 5 stages of prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis, which is pinching off of 2 things. Make sure you guys really understand the ins and the outs of this, because we will have lots of questions going on over this. Okay, so metaphase is the PMAT. Okay, let's go ahead and stop there because that's a lot of information, and I want you guys to be able to replay everything that happens in mitosis.